How do you prove the peptide inside a vial is exactly what the label promises? Not a look-alike, not a fragment, not a clever fake. For a small molecule, maybe you'd struck. For a peptide, the answer can decide whether a patient is treated or harmed. Hello, I'm Raghuvar Singh Parmar, Director Scientific Affairs at Resolve Mass Laboratories, and we work with pharma and biotech teams to confirm the identity of peptide and protein drugs and to catch what shouldn't be there. Today, we'll pull back the curtain on mass spectrometry and see how scientists actually prove a peptide is authentic. If you have found anything valuable in this channel, please like and subscribe. This gesture of appreciation means a lot to us. Picture a vial labeled semaglutide that turns out to hold something else. A slightly different peptide, an underpotent batch, or an undeclared impurity. As GLP-1 drugs went mainstream and demand outrun supply, counterfeit and research-grade peptides flooded in, and regulators began finding products that simply weren't what the label claimed. With a peptide, a single wrong amino acid isn't a typo, it's a different drug with a different safety profile. A label is just a claim. You know what's really in the vial, you need evidence, and that's the mass spectrometer. So, what does authentic mean for a peptide? More than people expect, it isn't enough that a peptide is present. Authentic means the amino acid sequence is correct, the mass matches the formula, every modification, disulfide bonds, end caps, attached chains is exactly as designed, and nothing undeclared is riding along. It's a fingerprint that has to match on every line. Miss one and you don't have the drug you think you do. Mass spectrometry is a rare technique that checks most of those lines at once. Here is the first thing a mass spectrometer does with your peptide. It weighs it, but not the way a scale would. It gives the molecule a charge and measures its mass to charge ratio. One peptide show up carrying several charges at once, and the software rebuilds those signals into one precise molecular weight within a few parts per million of theoretical. But the same mass can still hide a different molecule. Two peptides with the same amino acids in different order weigh the same. Matching mass is necessary but not significant. That gap is where fakes and errors live in three forms. First, the wrong sequence, the same amino acids in the wrong order weighing the same. Second, the wrong modification, sequence right but a disulfide bond and end cap or side chain is off. Third, hidden impurities, truncated or deletion peptides, shorter residue quietly riding along. Any of these can pass an identity test that only checks total mass. If this is sharpening how you look at your own methods, a quick like helps us. Reading the sequence, a peptide's real identity is its sequence, the exact order of its amino acids, and mass spectrometry reads that order by breaking the molecule apart on purpose. In second stage, MS, the instrument isolates the peptide and scatters it along its backbone into a ladder of fragments called B and Y ions. The gaps are the answer. The mass difference between neighboring fragments tells you which amino acid sits at which position. Read those gaps and the sequence appears. And because every fragment past a change is affected, one swapped amino acid shifts the whole ladder. So the wrong order fake that fooled the scale has nowhere to hide. Consider 3 real situations where a peptide looked right on the paper but was not. During the GLP-1 shortages, analysts flagged counterfeit vials that weren't the labeled drug at all. In synthetic manufacturing, deletion impurities, chains missing on one residue, sit so close in mass that only fragmentation separates them. And an epimer where one amino acid flips its chirality, carries the identical mass but behaves differently in the body. At Resolve Mass, we have had sponsors chase these signals, a peak, a hair off, a ladder with one wrong run, and turn worry into a clear identity call. Different failures, one lesson: read the molecule, don't trust the label. So, how does a lab build that proof start to finish? As a chain, prep and desalt so the peptide ionizes cleanly, intact mass to confirm the overall weight. For larger peptides, an enzyme cuts the molecule into predictable pieces, classic peptide mapping, MS/MS to confirm the residue order and pin down modifications, and orthogonal checks, amino acid analysis, chirality, and a hard look at impurities. Each step shuts a door a lookalike might slip through. Skip one and you have left a door open. Here is what to remember about proving a peptide is authentic. A label is only a claim, the spectrum is the proof. The right total mass is necessary but never sufficient, because a different molecule can weigh the same. MS/MS goes further and reads the sequence itself, fragment by fragment, and orthogonal checks, chirality, modifications, impurities, close the last gaps. That's how you turn it should be right into we have proven it is. If you are developing a peptide or protein therapeutic and want a CDMO or CRO partner who treats identity as the evidence to be earned, Resolve Mass Laboratory supports small and mid-sized pharma and biotech teams from early characterization through submission. Before you go, I want you to do 3 quick things. Subscribe to this channel right now. The more you subscribe, the more valuable pharma insights, expert tips, and industry updates we can bring to you. Please comment and let us know what did we do wrong and what did we do right until now. Because the more you give us the feedback, the better we'll be able to make videos for you. And share this video with at least one person who would find it useful. You never know who is stuck on exactly this kind of problem right now. I will see you in the next time. Until then, goodbye for now.